What if today we could peer 140,000 years into the past and uncover the true beginnings of Neanderthals and their uncanny kinship with us? In 1932, a peculiar child's skull was unearthed in Israel's Shrouded cave. Misclassified at the time, this skull is now believed to belong to a Homo sapiens Neanderthal hybrid, reaching across the chasm of species distinction far earlier than scientists ever imagined. This discovery rewrites the narrative. The paths of our ancient relatives didn't merely cross, they merged. Meanwhile, in remote Siberia, a fossil known as Denny, a child of a Neanderthal mother and Denisovan father, confirms that interspecies unions were far more common than previously believed. Jump forward to France, where a Neanderthal named Thorin survived in isolation, untouched by external gene flow nearly until the end of his time. And nestled in Central Europe lie the silent witnesses of admixture, the Zlati-Kuhn and Ranis genomes, bearing the scars of a single, defining mingling between Homo sapiens and Neanderthals around 45,000 to 49,000 years ago. This evolving story, etched into bones and genomes, reveals that the tapestry of human origins is woven from threads we share with the Neanderthals, rich, tangled, and astonishingly unexpected. For more than a century, the story of the Neanderthals has been told in fragments. Bones scattered across caves in Europe and Asia, stone tools buried in ancient sediments, and the faint echoes of DNA preserved in our own genomes. All these clues have offered glimpses of who they were. Yet, despite decades of study, the real origin of Neanderthals has remained elusive. Were they a distinct species born in isolation? Or were they, from the very beginning, intertwined with us? Our story and theirs inseparably linked. A discovery made in 1932 at Shrouded Cave in Israel is now helping to reshape that debate. At the time, archaeologists uncovered the partial skull of a child, but without the benefit of modern genetics, its features were difficult to classify. Some saw traits of early Homo sapiens. Others noticed the robust, archaic form more typical of Neanderthals. For decades, the find sat in collections, its significance overlooked. Recent reanalysis, however, has revealed something extraordinary. Using CT scanning and digital reconstruction, researchers found that the skull child bore a mix of features so unusual that only one explanation fits. This was likely the offspring of a Neanderthal and an early Homo sapiens. If true, it would represent the earliest known hybrid of its kind, dating back nearly 140,000 years, far earlier than the main wave of contact between our ancestors and Neanderthals in Europe. This child's existence suggests that the story of Neanderthal origins is not one of separation, but of contact zones, where populations met, mingled, and left descendants with complex identities. It also points to the Levant, the crossroads of Africa, Asia, and Europe, as a critical region where humans and their close cousins overlapped for tens of thousands of years. The skull child is only one piece of the puzzle. Other discoveries, from the Denisovan hybrid known as Denny in Siberia to the isolated Neanderthal lineage in France nicknamed Thorin, all add layers to a story that is far richer than once imagined. Together, these finds challenge our old picture of Neanderthals as a separate branch, and instead place them within a shared human saga, one that stretches across continents and deep into our genetic past. The Levant has long been recognized as a gateway between continents, a place where populations met and exchanged both culture and genes. Among its many caves, Skull stands out. Excavated in the early 20th century, it yielded human remains that were puzzling from the start. Some skeletons resembled early modern humans, while others bore archaic traits. At the time, the variation was explained as either evolutionary transition or simple population diversity. But modern technology has reopened the case, and the child's skull once buried in those deposits is now forcing a re-evaluation. High-resolution CT scanning and 3D analysis revealed that the skull child was neither fully Homo sapiens nor fully Neanderthal. Instead, the features fell neatly in between. A modern, human-like face, combined with a more archaic, Neanderthal-shaped brain case. This pattern is best explained by hybridization, suggesting that interbreeding between our species and Neanderthals occurred far earlier than the well-documented contacts in Europe, roughly 50,000 years ago. The implications are significant. If this child truly represents a first-generation hybrid, then the meeting of Neanderthals and Homo sapiens was not an isolated late event. It was part of a much deeper and older process. The Levantine corridor, situated between Africa and Eurasia, was a natural meeting ground where small populations likely encountered one another repeatedly over tens of thousands of years. Each encounter may have left behind genetic traces, some surviving into later populations, others vanishing with local extinctions. This challenges the tidy view of human prehistory, in which groups replace one another in succession. Instead, it paints a picture of overlapping populations, coexisting in the same landscapes, sometimes competing for resources, sometimes exchanging knowledge, and occasionally producing children together. The school child represents the human story at its most complex. Not a neat branching diagram, but a web of connections. Far from being an anomaly, this hybrid may have been part of a recurring pattern. And evidence from thousands of kilometers away, in a cave deep in Siberia, would later confirm that interspecies unions were not rare accidents, but a fundamental part of how human lineages interacted. In 2018, a tiny fragment of bone recovered from Denisova Cave in Siberia stunned the scientific community. Known simply as Deni, or Denisova 11, the specimen was initially unremarkable, a sliver of long bone, barely identifiable by sight. But when geneticists extracted DNA, the results revealed something unprecedented. This young individual, who lived about 90,000 years ago, was the direct child of a Neanderthal mother and a Denisovan father. This discovery provided the first clear genetic proof of hybridization between two distinct human lineages. Before Deni, researchers could only infer interbreeding from scattered traces in modern genomes. Here, however, was concrete evidence of a first-generation hybrid, someone who embodied the union of two populations once thought to have lived largely apart. Deni's genome told an even more intricate story. Her Denisovan father carried Neanderthal ancestry several generations back, showing that these lineages had crossed paths before. This suggests that interbreeding was not a rare chance occurrence, but a repeated part of human interaction wherever populations overlapped. Denisova Cave itself appears to have been a meeting place, with evidence of both Neanderthals and Denisovans occupying it at different times. What makes Deni so important is not just her hybrid status, but what she reveals about the fluidity of ancient human boundaries. Instead of sharp divisions between species, we see populations moving, meeting, and blending. To early humans, these encounters were not abstract questions of biology. They were lived experiences, where cultural exchanges, alliances, and family bonds blurred the distinctions drawn by modern science. The broader genetic legacy of these unions is still carried today. People of non-African descent inherit about 2% of their DNA from Neanderthals, while populations in parts of Oceania and Asia carry significant Denisovan ancestry. Deni is a reminder that these fragments of ancient DNA in our genomes come from real individuals, from children who lived at the crossroads of worlds. From the Levant's school child to Siberia's Deni, the pattern becomes clearer. Interbreeding was not an anomaly, but a recurring thread in human evolution. The next question is how these interactions shaped Neanderthal populations themselves, and in France, a discovery known as Thorin offers new clues. In the rolling hills of southwestern France, archaeologists uncovered a fragment of jawbone that would later be known as Thorin. At first glance, it seemed like just another Neanderthal fossil, important, but not extraordinary. Dating placed it around 42,000 years ago, a time when Neanderthals were approaching the twilight of their existence in Europe. But when researchers sequenced Thorin's DNA, the results told a strikingly different story from the hybrids found in the Levant and Siberia. Unlike Deni or the school child, Thorin showed no evidence of recent gene flow from outside populations. His genome was remarkably isolated, carrying signatures of inbreeding and limited diversity. This suggested that Thorin's group was part of a shrinking, fragmented Neanderthal population that had little or no contact with neighboring Homo sapiens or Denisovans. In other words, while some Neanderthals lived in contact zones rich with interbreeding, others remained in enclaves, genetically cut off from the outside world. This isolation had consequences. Genetic bottlenecks, the reduction of genetic diversity within small groups, are often linked with declining resilience. For Neanderthals like Thorin's community, this meant reduced adaptability to changing environments and increased vulnerability to disease and resource scarcity. In many ways, Thorin's genome represents the final chapter of the Neanderthal story. Small groups scattered across Europe, surviving for a few millennia longer, but with little hope of long-term continuity. Thorin's discovery highlights the unevenness of Neanderthal history. They were not a uniform population experiencing the same fate everywhere. In some regions, they mingled and exchanged genes with newcomers. In others, they persisted in isolation until their line faded away. This patchwork of outcomes complicates the old narrative of Neanderthals as a single, cohesive population wiped out by the arrival of Homo sapiens. Instead, their decline was uneven, shaped by geography, contact, and chance. By the time Thorin lived, hybridization events were still occurring elsewhere, but his lineage seems to have been cut off from them. His DNA provides a rare glimpse into the last stages of a population approaching extinction, isolated, vulnerable, and yet still distinctly Neanderthal. For decades, scientists debated when our species first encountered Neanderthals. The classic view held that Homo sapiens left Africa in a major wave around 60,000 years ago, entered Eurasia, and soon after, interbred with Neanderthals. This was thought to explain why nearly all non-African people today carry about 2% Neanderthal DNA. But recent studies are reshaping that timeline. Genomes from early Homo sapiens fossils in Central Europe, such as those from Zlatikun in the Czech Republic and Ranis in Germany, show evidence of contact with Neanderthals as early as 45,000 to 49,000 years ago. This fits with archaeological findings of modern human tools spreading through Europe around the same period. These individuals, sometimes called the first Europeans, were part of an early wave of Homo sapiens moving north from the Levant. Their genomes reveal long stretches of Neanderthal ancestry, a hallmark of recent interbreeding. The fact that this Neanderthal DNA appears in all modern non-African populations suggests that a single major episode of admixture occurred shortly after humans left Africa, before their descendants dispersed across Europe, Asia, and beyond. In other words, the interbreeding event was not repeated endlessly in every region. It happened once, early, and then spread with migrating human populations. Yet, this does not mean contact was rare. The schoolchild in Israel and Denny in Siberia prove that interbreeding occurred at multiple points and in different regions. But only one of these events left a genetic signature that persists widely today. Others were absorbed into local populations that eventually vanished, leaving no descendants. This revised timeline complicates our understanding of the out-of-Africa migration. Instead of a single, clean departure, it seems there were waves of dispersal, some as early as 200,000 years ago, where small groups ventured out, encountered Neanderthals, and sometimes blended with them. Most of these early ventures ended in extinction, but the genetic traces they left behind provide a ghostly record of those forgotten journeys. The story of Neanderthal admixture is not just about who mated with whom. It raises a deeper question. How did these genetic exchanges shape the biology and adaptability of the populations that followed? That question leads us to the role of hybridization in survival itself. Each new fossil, each newly sequenced genome, seems to pull apart the tidy timelines we once relied upon. For most of the 20th century, the story of Neanderthals was framed as a simple progression. They emerged in Europe, thrived for hundreds of thousands of years, and were replaced by Homo sapiens around 40,000 years ago. Interbreeding, if it happened at all, was considered rare and insignificant. But discoveries like the schoolchild, Denny, and Thorin have forced a rethinking of that narrative. We now know that Neanderthals were not merely a European phenomenon. They were part of a wider network of populations spread across Eurasia, overlapping with both Denisovans and early Homo sapiens. Evidence of contact stretches back at least 140,000 years, meaning that our lineages were entangled for far longer than once imagined. Instead of two species meeting only at the end of Neanderthal history, their interactions were woven throughout their existence. The revised timeline also shows how uneven these encounters were. In some places, like the Levant, early Homo sapiens entered repeatedly, sometimes leaving descendants who interbred with local populations. In others, such as parts of Western Europe, Neanderthals remained relatively isolated, like Thorin's group. Rather than a single global event, the story is one of regional variation, with different outcomes depending on geography and circumstance. This new framework reshapes how we think about human evolution as a whole. No longer is it a tree with clear branches that split and never touch again. Instead, it looks more like a braided river, channels diverging and reconnecting, carrying sediments of ancestry that blend and disperse. Hybrids like the school child or Denny are not exceptions to the rule, but reminders of how porous the boundaries between populations really were. The shifting timeline also reframes the end of the Neanderthals. Rather than being suddenly replaced, they were gradually absorbed into a larger human population. Their disappearance was not an extinction in the absolute sense. It was a merging. Their genetic legacy carried forward within us. The question now is, what did this blending mean for us biologically and culturally? To answer that, we need to examine how Neanderthal DNA influenced survival, adaptation, and even aspects of modern human life. When we talk about interbreeding between Homo sapiens and Neanderthals, it's easy to focus on numbers and percentages. The 2% of Neanderthal DNA carried by non-African populations, or the varying amounts of Denisovan ancestry in Asia and Oceania. But these genetic fragments are not abstract. They shape the biology of our ancestors, and in some cases, continue to affect us today. Studies of modern genomes have revealed that Neanderthal DNA influenced traits ranging from immune responses to skin and hair characteristics. For instance, certain Neanderthal gene variants improved resistance to pathogens, giving Homo sapiens an advantage as they entered new environments. Other variants influenced keratin production, which may have helped early humans adapt to colder climates by strengthening skin and hair. Yet not all inherited traits were beneficial. Some Neanderthal-derived genes are linked with conditions like type 2 diabetes, depression, and nicotine addiction. Hybridization, in other words, carried trade-offs, advantages in survival alongside lingering health risks. The cultural implications are harder to trace, but equally important. Archaeological evidence shows that Neanderthals were capable toolmakers, producing stone implements, pigments, and even symbolic artifacts. Contact with Homo sapiens may have facilitated exchanges of knowledge and techniques, blurring the lines between what each group invented. In some sites, tool traditions once attributed solely to modern humans now appear to have been shared or parallel innovations. The boundary between Neanderthal and Homo sapiens culture, like their biology, was porous. The presence of hybrids also reminds us that these encounters were deeply human. The school child, Denny, and others like them were not just genetic junctions, but members of communities. Their existence suggests relationships, families, and social networks that spanned what we call species. While we may never know the cultural contexts of these unions, whether cooperative, opportunistic, or rare, they testify to a level of interaction more complex than simple competition for resources. Far from being evolutionary dead ends, Neanderthals contributed directly to who we are. Their DNA is woven into the very fabric of our biology, influencing our resilience, our vulnerabilities, and perhaps even aspects of our behavior. To fully understand the legacy of Neanderthals, we must place them not outside our story, but inside it, part of the continuum of humanity. The search for the origin of Neanderthals has brought us to a surprising realization. Their story is inseparable from our own. Once imagined as distant cousins who lived apart and disappeared without a trace, Neanderthals are now recognized as contributors to our ancestry, our biology, and our shared history. The evidence tells us that encounters between populations were not rare accidents, but recurring events. From the Levant's school child to Siberia's Denny, from the isolated Thorin lineage in France to the early admixture recorded in Central Europe, each discovery reinforces the idea of a braided human past. The boundaries between groups were never absolute. Populations moved, met, interbred, and sometimes vanished. But in the process, they left behind threads that became part of us. This new perspective reshapes not only our understanding of Neanderthals, but of evolution itself. Instead of a clear-cut sequence of species replacing one another, we see a dynamic web of contact zones, exchanges, and absorptions. Extinction, in this sense, was not the end of the Neanderthals. Their genes live on in billions of people today, influencing traits as intimate as how our immune systems work or how our bodies adapt to climate. The broader implication is a humbling one. The story of human origins is not about purity or separation, but about connection and mixture. Every fossil and genome we uncover points to the same conclusion. Human history has always been intertwined. Our survival was not achieved alone, but through interactions with other hominins who shaped us in ways we are still uncovering. As new technologies continue to extract information from ancient DNA, more surprises will come. Perhaps other hybrids remain to be identified, hidden in overlooked fossils or unexamined bones. Each will remind us that our family tree is more tangled, more interconnected, and more human than we once believed. The Neanderthals did not simply vanish. In a real sense, they endure within us.